So now we've finished our 3 types of enzymes, and we've done 2 out of the 3 types of processes that we need to understand with DNA manipulation. So we've talked about restriction endonucleases, we've talked about ligases and DNA polymerases as our 3 enzymes. We've also talked about bacterial transformation and PCR as 2 out of our 3 processes. So we have 1 more to go. So gel electrophoresis, I've actually broken up into a series of different videos where we talk about what and why, then we talk about how you do it, and I'll go through a online simulation as part of that, just because some of you might not have Flash, or you might want to go through with me how much you want to write down. But essentially that's what we're going to be going through. Then we're going to look at how it works and what factors influence it working, some examples, and then finally the ladders and the standards. So we're going to do quite a bit of work on gel electrophoresis. It's really cool, and this is one of the techniques that we get to do in class, so we're going to get lots and lots of practice to do with electrophoresis. And then that will close us off for the end of the DNA manipulation bit. So to begin with, we need to talk about what is gel electrophoresis. It's a big word, and why do we care what it can do for us? So the first thing is, let's think about it. Gel, meaning it's going to have something to do with gel, so we make an agar-like jelly. And also electro means it's got something to do with electricity, and the phoresis part is talking about the separation process. So it's using gel and electricity to separate out things. In particular, we talk about separating out DNA is our main one, but it can also separate RNA and proteins. So any of those biomacromolecules that have a charge. So thinking way back to our structure of DNA, you might remember that the backbone, the phosphate group on the backbone, actually has a negative charge. So DNA on its own is negatively charged. We can use that property to separate it out into different sized fragments. DNA is tiny. There's no point in trying to do this with a microscope. So what we do instead is we use a process called gel electrophoresis to separate out different sized fragments of DNA. And you will see this, it makes so much sense when you do it. And I'm hoping that you really enjoy this process as well. So we can separate DNA fragments and proteins, but mostly we talk about DNA according to their size and their charge. Why would we want to do that? So what is the point? Why do we care about separating out different pieces of DNA? So remember, when we use a restriction enzyme or a restriction endonuclease, we just cut a piece of DNA. So that's all happening in a tube, and we don't know where the bits are. You can't extract them using a microscope. You can't just go, oh, I'm going to take the little bit here. So say you want that insulin gene. So you've taken your human chromosome and you've cut it up with restriction endonucleases. How did they actually take that insulin gene and not just take all of the mishmashed DNA that was there? So that's what we use gel electrophoresis for, separating out those pieces of DNA so we can use them the way we want. There are some other reasons. If you have a particular section of DNA and you cut it with restriction enzymes, every different piece of DNA will cut at different places. So if you have a length this long, you might cut it here and you get 2 fragments. Say you have a length this long from a different person, maybe the restriction enzyme is going to cut it twice and you'll end up with 3 fragments. So what we need it for is we need it to separate out different fragments. As a result of that, because my DNA is not the same as your DNA, each one of us leaves behind a DNA fingerprint. So they don't just zoom in on your DNA and sequence the whole thing. This is a very cheap, relatively cheap, easy way to create a profile for someone. Although they have removed DNA profiling from the course for 2020, we haven't removed it from this sense. So I'm still going to talk about it. And although you shouldn't be asked questions on it, it's really hard for me to teach this unit without talking to you about DNA profiling. So we use it to establish crime scenes. We can also use it for paternity testing because you can figure out whether or not the child's DNA matches the mother's or matches the father's. Whether or not she should match the mother's, but whether or not who the father is by looking at the DNA fingerprints of the two. You can also look for evolutionary relationships between organisms, depending on how similar two organisms' DNA is. You can also test for genes associated with particular diseases if you know the lengths of DNA that should be coming out from a particular digest, which is DNA mixed with a particular restriction enzyme. If you know that you get a certain pattern with a unaffected gene and a certain pattern with an affected gene, you can actually do that really easily and see whether or not someone has the gene for a particular disease. And the final one is the one that I talked about to begin with. We can use this to isolate genes like the insulin gene for doing bacterial transformation. So here's just a really quick picture. I'm going to come back to it later on. But essentially, you load your mixture of macromolecules up the top here. So this would be your DNA, your cut up DNA fragments. So this is when you've got DNA, you've mixed it with a restriction enzyme, and now you've got DNA of different lengths. You've got this porous gel. You run electricity through it, and you end up separating them out. So you've got your big yellow ones at the top, your medium-sized green ones in the middle, and your small red ones down the end. And we're going to spend lots of time breaking down what this means.